A warm good afternoon to all of you who participants and today I am introduce today's guest speaker. I am Anand, working as assistant professor in Sri Vishnu College of Pharmacy. So it's my privilege to introduce today's our speaker, eminent scientist, Dr. K. Suresh Kumar Garu. Uh, So presently, Sir is working as a principal scientist at Center for Natural Products and Traditional Knowledge, CSIR, Indian Institute of Chemical Technology, Hyderabad, India. So Sir has a professor at Academy of Scientific and Innovative Research and Education and Research background. Sir completed his PhD from CSIR IICT in 2005 on. So, her conducted postdoctoral research at the National Center for Natural Products Research, University of Mississippi, USA. So, Sir has specialized in natural products and medicinal chemistry. So, Sir research interests are discovery and development of small molecule immunomodulators derived from natural products, standardization and validation of commercially significant herbal drugs. Sir, investigation of bioactive natural products for pharmaceutical applications, particularly in immunology and anti-infective drug discovery. Sir, contributions and achievements are around 215 research publications in reputed and international journals. Under H index, Google H index is 34 and his citations are around 4,400 plus. So at high impact factor journals. Supervised and mentored numerous PhD scholars and postdoctoral researchers, principal investigators in several drug discovery projects. Sir got an award as an Young Scientist Award in 2010 from OPPI, Organization of Pharmaceutical Producers of India, and also from Andhra Pradesh Academy of Sciences. Sir recognized his significant contributions regarding natural products and medicinal chemistry. And Sir associated with various national and international scientific committees, members of leading scientific and organization in the field of pharmaceutical sciences and natural products and chemistry. Sir, good afternoon, sir. I'm welcoming for the day 5 of FDP. Thank you, sir.
Yeah. Good afternoon. Am I audible?
Yes, sir.
Yes, sir.
Okay. Thank you. Thank you, sir, Natras sir, for giving the opportunity to share our recent research results and to the platform, FDP platform. Just as you are aware that I have been working in this area of natural products and medicinal chemistry and also some herbal drug standardization using the HPTLC and LCMS. So today I would like to enlighten about the various recent approaches in natural products research for drug discovery. So I will also share some of the recent research results by the application of these approaches. So first basically traditional medicine is the base for many natural products. Traditional medicine basically evolved through the scientific knowledge or medicinal knowledge from indigenous community or local community around the world. So in those ancient days, if the people used to identify the surrounded plants as edible plants, medicinal plants, and poisonous plants. So this knowledge has been rooted to their system of their entire system, and that has been became converted as a traditional system of medicine. As we can call upon Indian system of medicine. Like every country has their own system of medicine. and this is a knowledge developed from one generation to other generations. And in India, particularly, the traditional medicine can be classified into two categories. One is codified and non-codified system. Codified will come under Ayush Ministry like Ayurveda, Unani, Siddha, and homeopathy, where there are some treaties. Based on the treaties or pharmacopoeias, this will be practiced. But where coming to non-codified system, it's called as a folklore medical system. And there is no scientific basis, but even till today, the majority of the tribal people are practicing this folklore medicine system, where they got their knowledge from their forefathers and ancestors. Of course, when coming to the contributions of these two systems, in India particularly, they are equally contributing for the primary health care in India. So if you see this kind of traditional system where more than 5,000 years, 4,000 years it has been serving, but the only hurdle or the problem is quality control. But in this era, we can we can come across the quality control by the applications of several scientific techniques like validation of herbal drugs by using hyphenated techniques, biological validation. So we can do much better. And if you see the modern countries also, developed countries also, the traditional medicine is in practice, but of course it is a different name. In America, those are called botanicals. In Europe, those are called nutraceuticals, but the raw material is same plant medicinal plants. And these are some of the over-to-the-counter shops available in the different countries. And this kind of people we can come across when you visit the tribal people or interior forest where there is no access of medicines as well as the access of no doctors. So these people are called traditional healers or traditional practitioners, and these people are completely completely based on their healthcare, based on their folklore medicine system, and they also serving the surrounded villages for the particularly in the critical cases, and they are doing much better. Recently, World Health Organization has conducted a survey. Central Government of India conducted a survey regarding the existence of these traditional healers. Almost 4 to 5 lakh people are existing in the entire country, in every corner of the country, and they got their knowledge from their fathers and forefathers, and they are doing much better, even though they don't have any degree, but they know how to prepare the formulation and how to prepare and how to advocate what is the dosage, everything they know. So that's what recently Government of India also thinking to identify a few more people and give for their recognition. And based on these peripheral results, recently World Health Organization conducted a survey regarding the existence of regarding utilization of the plant-based medicine. It revealed that more than 80% of the world population depend upon the plant-based traditional medicine for their primary health care, and also further listed another 21,000 medicinal plant which has been reported huge medicinal values around the world. And when coming to India, India has 16 agroclimatic zones, around 45,000 plant species, and has rich medicinal flora, and majority of the flora were part of our system of medicine, like traditional system of medicine like Ayurveda and Siddha Unani. And further another 1,500 medicinal plants were identified, and some of the species are also fairly exporting to other country, and currently another 5,000 products were included in the pharmacy of Ayurvedic treatment. So if you see, these are the major natural resources like plant kingdom, marine world, animal sources, marine and venoms and toxins, and the microbial world. Among these 5 sources, plant kingdom has served a lot in the form of the Ayurveda and Siddha and other homeopathy medicines. And microbial world, coming to microbial world, the penicillin is one of the best example where penicillin was discovered from the microbial source. And the animal sources, there are some drugs also available in the present market which has been discovered from animal sources. Best examples are epipatidins discovered from the frog. And similarly, marine world, there are a lot of antibiotics and anti-cancer agents which are present in the market are derived or originated from the marine world. Similarly, some of the drugs also available are discovered from venoms and toxins. And these are the different applications of natural products. Natural products applications are everywhere. They have a wide range of applications in each and every field, starting from herbal medicines, food and feed, pesticides, cosmetics, and pharmaceutical applications in each and every field. And I just quoted few examples where there is a lot of highly where these are very part of major industries like beverages industry. Epigenin is one of the major compound used in the beverage industry as a colorant. Similarly, linalool used as a perfume in the perfumer industry. On the stevia side, we are very much well known about the stevia side, which are useful as artificial sweetener, and artemisinin. And for the discovery of artemisinin, no Chinese professor got Nobel Prize also. Similarly, there are a lot of other compounds also there which has been extensively applied in different fields. And recently, the survey conducted by the Indian universities regarding the consumption of the Indian spices, particularly, we generally use spices in every day-to-day life, starting from morning to evening. We consume a lot of spices like ginger, garlic, ilachi, and turmeric, and different spices we consume. So based on the survey, what they found is the persons who consume lot of spices will have a lot of immunity. So that indicates our spices are major sources for type of compounds like polyphenols, and the polyphenols are known for their adjuvant properties. So adjuvant means the compounds which increase the immunity response are called adjuvants, which are the immunity responses. So that's what after COVID period, majority of the people are now taking or consuming lot of spices, and that shows the importance of the spices. So why herbals means, as I mentioned there is a long history of utilization of these or consumption of these herbals from the ancient times to till date, and in India it can be traced back to 2600 BC, where in the Mesopotamian civilization, and in Egyptian it is also traced back to 1500 BC, and in that period more than 700 drugs were documented in the Egyptian civilization. And particularly when coming to Chinese materia medica, which is a famous system of equally important system of medicine like India, it is also traced back to 1100 BC. And these are traditionally established by the Ayurveda and other systems. As we know, Ayurveda is a long established system of medicine, and this is also getting attention worldwide as an alternative system of medicine. Even the Ayurveda speaks about the holistic medicine, but majority of the drugs which are in the present market were derived based on the Ayurvedic knowledge. Here I just quoted few examples of the drugs, Rawalpindi alkaloids, which are derived from Rawalpindi serpentina. Similarly, soralins used for vitiligo, derived from Soralia corifolia. Similarly, Olar henna alkaloids, which are very much used for hemibiasis, and L-Dopa, derived from Euchinum purensis, are very much famous for Parkinson's disease. Similarly, pepperidins are known for bioavailability enhancer, derived from different pepper species, and philandis, curcumins, vithanilides. These are all very famous drug lead compounds derived from based on the Ayurvedic knowledge, and these are all economically affordable, as some of the medicinal, these plants are also cultivatable, not only wild source, and these are comparatively low toxic, but it has to be taken the optimum dosage, then only these are low toxic and also very effective in the chronic diseases. So if you see the contribution of natural products from the last 4 decades, and almost 1200 marketed natural product drugs were marketed for different diseases, and major contribution goes to like hypertension drug and anti-cancer drugs and antifungal drugs. Here, this iceberg shows only 10% of the flora has been worked out so far. And the remaining 90% of the flora is not at all explored. And this also shows one of the good opportunity to work on this field. When coming to our own contribution, our own research in the area of natural products, we are working in different like basic and applied research. In basic sciences, as I mentioned, we are very much interested in the working in the natural products, and we regularly interact with the tribal people where to understand their knowledge. And after interaction, we come to lab, and then whether we will search in the literature whether those plants are scientifically validated or not. If it is not validated, and we will take up the challenge to identify the active compounds or active fractions. And after identification of the active compound also, we'll also do some kind of medicinal chemistry to enrich its activity and reduce its toxicity. And here, and we are working on different targets like anti-cancer, antifungal, and anti-diabetic and free radical scavenging activity, and agriculture side, mosquito-side, insect side. And coming to applied side, we are actively involved in the standardization of the multi-herbal formulations, and also working on the process development of active phytochemical ingredients, where there is no need of doing any column chromatography. This process is which is viable to the many of the herbal industries, and also involved several other natural products projects. So if you can look at the approaches in the natural product research, natural products research drug discovery, there are different approaches starting from bioassayer approach and random approach and the utilization of the hyperrated techniques and phytopharma. So the bioassayer approach is the very famous one, and majority of some of the drugs also being discovered by using the bioassayer approach. There are different stages involved in the bioassayer approach drug discovery, and like extraction and screening of the various targets, and fractionation, screening of the fraction for different biological activities, and taking the active fraction, isolation, identification of bioactive constituent from the active fraction, and that compound should be characterized by the 1D and 2D NMR technique. Similarly, when coming to random approach, this is completely opposite to the bioassayer approach, because sometimes what happens is in the bioassayer approach, the active compound is very less in quantity, and if the phytochemical marker, which is the major compound, is high, major in quantity, and which is, if it is not active, the phytochemical marker, sometimes there is a possibility of masking the active compound by the phytochemical marker that is a non-active compound. In that case, we may miss that active compound. That's what we are also practicing the random approach, where you do extract with different solvents, non-polar, and isolate the compounds as many as you can, and finally screen for the different biological targets. And when coming to third approach, hyperrated techniques, nowadays hyperrated techniques are very much played a vital role in the drug discovery as well as dereplication techniques like LCMS, UPLCMS, MS, and molecular networking approach. This will reduce the time as well as increase the efficiency, and also the accuracy is also very more by using the hyperrated techniques. And phytopharma approach, this is a recent approach where no need of doing any purification like a single compound. It is like a extract or fraction, which should contain minimum 4 number of markers, and that 4 number of markers are qualitatively and quantitatively identified and characterized. If it is that also should be standardized with different standardized in those markers, 4 markers in the extract or fraction, and those markers should be well characterized by using NMR and 2D NMR techniques. Further toxicity and all should be done. Then you can file a patent or you can introduce the product also. This is a phytopharma approach. And coming to, so I will give some of our recent examples where we followed the same. These strategies. First, bioassayer approach. So we have developed a bioassayer approach by simple modification. As you take the crude extract or active extract and pass it into the reverse phase 18 column by eluting with methanol and acetone, aqueous methanol and acetone to generate the 6 fractions. And all the 6 fractions were subjected to the biological screening, and the active fraction will be taken up for further isolation of the active compound. And once after obtaining the active lead compound, then it will be subjected to synthesis, because for doing the in-depth biological activities, multi-gram level compounds should be needed, and in vitro, in vivo studies, mechanism of action studies, and chemical modification. So I will give some of the examples where we have used the bioassayer approach for the isolation of the compounds. So here, the piper species, we know pretty well about the piper species, because piper nigrum is the one of the species fruits where we consume daily in our which is existing in our kitten, and we consume a lot. And as part of our program, we just working on different piper species, and we completed almost 7 to 8 species of piper langum, piper nigrum, piper chaba, and piper beetle, piper attorney atom like. So here by using biosecond approach, we have screened the paper nigrum methanolic extract and piper chaba chloroform extract and piper chaba methanolic extract. Here, the piper chaba methanolic extract showed potent anti-cancer activity, and piper chaba chloroform extract showed moderate aldose reductase inhibition activity, that is diabetic retinopathy. So, based on this active extract, we subject to this extract was subject to the biosecond approach to isolate for the isolation of the compounds. And here we have isolated the 7 compounds from the active extract of piper chaba, and again these 7 compounds were subjected to the anti-cancer activity pure compounds, and one compound 2 is showing good activity with almost equal to the doxorubicin, again colo 205 cell line, and the among these 4 compounds were new compounds, others are known compounds, and these are the compound structures we confirmed with the help of different NMR and proton and C30 NMR and different 2D NMR techniques, HMBC, HSQC and COSI. Finally, we confirmed the structure by using the mass fragmentation studies, where we came, which is a dimeric piperamic alkylate, is forming the Diels-Alder reaction of monomeric piperamides like piperin and fragamide. So similarly, the active piper chaba extract, chloroform extract, also subjected to column chromato biosecond approach and isolated different compounds among these various class of compounds, including lignans and amides. And among this pipelartin, IC50, pipelartin is we got as a major compound, and it is showing moderate activity against AR enzyme with 160 micromoles. 160 micromoles. And since we got the major yield, and we thought of making the analogs of this compound, so before that we just analyze the structure with the marketed drugs, where we came to know this diamide functionality seems to be common in our compound as well as the marketed drugs. So we thought of keeping intact of this the functional group diamide functionality and playing around this aromatic scaffold and this double bond. For this, initially we analyzed the Michael-Addition reaction of pipelartin by introducing different indoles. As we know, this indole is one of the important heterocyclic moiety, which is a part of majority of the drugs in the market. That's what. For this, initially we just optimized the reaction conditions by screening the different Lewis acid catalysts. And here the iodine 10 mole percent is showing giving the good yield, 80% yield, and solvent dichloroethane is giving almost 80% yield. With this optimized condition, we prepared the Michael-Addition, monomichaelites and dimichaelites. These are the different types of compounds prepared by the Michael-Addition reaction of indole with pipelartin. And here you can see dimichaelites 3C and 3E showing good activity, almost 4 micromoles, which is almost equal to the standard drugs. That's what. No, the parent compound activity was 160 micromoles, and the derivative activity was 4 micromoles. So that means the activity was increased almost 40 folds. So similarly, we also prepared the different kinds of drugs and different kinds of modifications and prepared a lot of derivatives. Unfortunately, these compounds did not show any activity, and still we are working again by introducing a different pharmacophore to the pipelartin. Similarly, this is another project where we also followed the same biosecond approach. Praemana lardifolia, which is a part of the famous formulation called Dashamolaristum. This plant is also not validated scientifically. That's what we taken this plant and then subject to the biosecond approach and identified 4 new compounds, unique type of compounds, and these compounds were also screened for the anti-cancer activity. Almost all activities are on par with the doxorubicin, but the problem is these compounds are very scarcely available in the nature or in the plant. So almost we started with raw material, 10 kg raw material, we ended up with the 2 to 3 milligram. So which is not sufficient for doing the in-depth biological activity and validations. That's what we also prepared some synthetic strategy to get the gram level quantity of these compounds to elucidate its mechanism, how this is working as anti-cancer activity and all those things, which is in the process. And the literature says the tricyclic core of isotexate diterpenes are, this is a more important of the tricyclic core, which is the responsible for its anti-cancer activity. So these are some of the other examples where we also followed the biosecurity approach strategy for the isolation of these compounds like Premna tomentosa and like Doris candens and Hedychium spicatum and Plumbago capensis, and also some compounds from different neem oil and some Virginia or Mallotus philippensis, and similarly and other compounds also we have isolated Hedychium spicatum. We isolated the lead compound and then by using biosecurity approach and then made some of the chemical modification to increase its activity. So in majority of the cases, we observed the derivatives activity was far better when compared to the parent compound. And these are also some examples. And the next, coming to the reverse biosecurity approach or random approach. So as I mentioned, this is also equally important approach for the discovery of the new compounds or new bioactive compounds from the natural products. So here we have taken chosen this plant Cypadessa bacifera for this random approach. This plant is taken with the interaction with the tribal people from the Rampachodavaram and the Araku areas, where we came to know these plant extracts they are using for their small infections like antibacterial and other infections, and also they are also using this extract for the application of as an insecticidal agent, like mixing with monocrotophos and spraying the different agri fields like a paddy and other fields. So which attracted our interest to work on this plant, and then we have prepared different kind of extract like chloroform extract and methanolic extract, and we have isolated almost 30 to 35 compounds of almost biosynthetic sequence compound, and all these compounds were screened for anti-cancer activity, and almost all compounds were showing moderate activity against the different cancer cell lines. So these are the structures of the compound and activities. And here the Cipaferin G we have isolated almost 300 to 400 milligram, and by using this we just did the deesterification and then enclosed some the pharmacophore group, which really rewarded the increased its activity almost against the HEPG2 cell line, and also it also showed 100% inhibition against the antifutant activity against the Spodoptera litera at the 100 microgram per centimeter concentration. So these are the other examples which we followed the same random approach from the Schisandra grandiflora, where we have isolated the different kinds of sesquiterpenes from the non-polar extract, and from the polar extract we have isolated the different polar compounds like lignans, and by use which is showing very good, almost equal to the doxorubicin 0.24 micromoles. And we also studied the mechanism, and these compounds are the working by the tubulin polymerization inhibition, and we also did and this following the tubulin inhibition polymers, and did the molecular binding studies also. Similarly, recently we also worked on some plant Leonatis nepetifolia, which is the part of the folklore medicine in the Chhattisgarh area. Tribal people are using this, and we have taken this plant for our study and isolated one new compound which is showing the moderate activity against the cancer cell lines, and the one compound isolated the nepetifolinol 2 compound in good yields, and then we made the some of the derivatives by the modification of the different regions lactoning and phenolic group, and prepared the different derivatives. Among these compound 15, one of the acetylene derivative, triple one derivative, is showing almost good activity against the cell line, HeLa cell line, and this was studied further to know the mechanism of action, and also molecular modeling studies also been studied to identify the binding, which is binding at the colchicine site, and this work is also patented. And we are also working on the marine products like a marine organism like a seaweeds. We have identified, isolated by some of the suspend spartan diterpenoids and dolobelin diterpenoids by using the random approach, and the majority of the compounds are showing a very good anti-cancer activity. And further, we also studied the cell cycle analysis above this pathway on different staining assays to elucidate its mechanism, how it is acting as anti-cancer activities, and these are unpublished results. And as I mentioned, we were also recently developed a process for the active isolation of the active phytochemical ingredients without doing any purification or column chromatography. So this is one of the example where we have isolated the beta amarin from the crude extract, and after making the enriched extract fraction, and just we washed it with the different solvents, and we got the almost 99% of pure beta amarin. If you see the cost of 10 mg beta amarin cost about 64,000 in Sigma reach, and we have isolated 6.2 grams of beta amarin and from the 2 kg of material. So that is the beauty of process development in the natural products. So once we got the good quantity of this beta amarin, we also did some of the medicinal chemistry approaches to generate the different heterocyclic analogs or scaffold fused beta amarin derivatives and screened for the anti-cancer activity, and majority of the compounds showed good activity. And here again, we are also working on different the waste to wealth concept. And here we have taken the fruit peel of the Garcinia mangostana fruit, which is called mangosteen. And the pulp is very nutritious and very good, having a very good antioxidant, other nutritional properties, where the pericarp is like a used by the consumed by the some of the tribal people for different virus diseases. And we have collected this the pericarp of this fruit and isolated different xanthone compounds and tested for chikungunya against the chikungunya virus. And the mangosteen, the second number 2 compound, is showing very good activity. And based on this, we also made different kind of derivatives, and almost all derivatives, majority of the 80% of the derivatives are showing on par with the more activity, more antiviral activity than the mangosteen. And this work is also recently published, and also we got a patent for this. And again, these are some of the examples where we have used the random approach for the isolation of these compounds. And we also did some of the derivatives and identified, and some majority of the derivatives showed very good activity. These are some other compounds isolated by using a random approach. And the lichens, I think this is also one of the natural source where lichens also known for their properties from the ancient times. But so far there is no drug has been discovered from the lichens, but some of the lichens were used or added into the some of the drugs as an cosmeceutical and nutraceutical drugs. And lichens are nothing but symbiotic organism of fungi and cyanobacteria. And majority of the lichens are containing the polyphenolic compounds, which are very well known for antioxidant activity and alpha glucose inhibition activity. And these lichens we consume, majority of the lichens are edible. For example, if you buy biryani material in supermarkets, like it also contains the part of lichen along with your dalchini and cinnamon and other biryani leaf. So that's what the lichens are not explored chemically. So we are also working in this field. Lichens also called ecological indicators. Here, this is a lichen Usnea longissima, which is called old man beard. And this lichen was fractionated by using hexaethyl acetate and then purified with the different by the gradient system of hexaethyl acetate to get the 4 compounds. These are the structures of the 4 compounds. And first time we developed the crystal structure of barbatic acid, compound 21. And we also screened these compounds against the anti-cancer activity, and almost barbatic acid showing good activity against the A549 cell line. And further, we also studied the mechanism of action cell cycle studies. And in cell cycle studies, we came to know this the cell cycle in G0 by G1 phase leading to the apoptic cell death, and further staining assays and other also indicated that the immunoblast assay also indicated this is induces apoptosis by robust cleavage of the PRP protein. Further, we also taken the help of hybrid techniques to identify the compounds in picogram level by developing a total ion chromatogram of crude extract, and identified the character, the structures based on the fragmentation, identify the fragmentation and the characterization by using the mass spectrometry principles. And we also further justified how this compound is forming by taking the help of the MSMS and fragments, and which is the key fragment for the formation of the compounds, also identified. from the mass spectrometry, UPLC, MS. And when coming to LCMS guided approach, this is a recent approach where we use the hyphenated techniques for the isolation of the compounds. And this is the traditional procedure for the isolation of the compounds in the natural products area, where you do the extract and then do chromatography for getting the fractions, and then bioassay to pick up the active fraction and purify it. And again, to get the pure compounds, and pure compound, again, you need to do the structural isolation and activity confirmation. And if it is a known compound, then you will be disappointed. And if it is a known compound, you are excited. But however, it takes a long time for starting from extract to pure compound or activity new compound. So to reduce this process, hyphenated techniques have played an important role on like a UPLC, MS or QT of MS and GCMS. So here what you do is you just prepare the extract and inject into the QT of MS, and you will get the molecular weight and molecular formula of each and every compound. And if it is connected to the different databases like SciFinder, Dictionary of Natural Products and Reactions, you can verify their identity, whether those are known compounds or new compound. If it is a new compound, just purify it, pinpoint that new compound and do fractionation, and then purify it and be subject to the activity. And here we follow the same thing. For the Trichilia conrads, same, this plant also taken with the interaction of the tribal people from the Rampachodavaram region. And this plant is also belongs to neem family, Malaceae family. What we did is here we prepare the extract and subjected to the and generated the total ion chromatogram of the extract, and then by taking help of Dictionary of Natural Products, we have identified 3 new compounds in the extract, and then these 3 new compounds were fractionated by using the preparatory HPLC, and then further purified to get the pure compounds, and then getting the NMR data, and we confirm the structure. These are the pure compounds structures, and for a glimpse I gave here proton and C13 NMR of compound 1. So these are all new compounds, and again, this is another example of by taking the help of LC, MS, HRMS approach for the isolation of these compounds from the lichen called Opioforma ventosa. This reticular pigment was taken and then isolated these compounds by using the LC HRMS approach. And we also used this LC-MS for in different type of applications like metabolite profiling and the quantification of different compounds from the same genus belong to of different species, and like a UPLC, MR, multi-reaction monitoring method quantification. And if you see the details, this is the main main objective is to develop a simple method for the quantification and simultaneous determination of the compounds from various species. And this is a method of choice for quantification in MS. And MRM method is a very sensitive and highly specific because at the same time it is analysis of parent and fragment ions that are characteristic of target molecule, and it is extensively used in pharmacokinetics and metabolomics. This is the first of its application in the natural products, particularly in the lichens area. So in the study for MRMSA study, we have taken 5 lichens, Permetrema tinctorum and Permetrema prasodianum, Permetrema ornitum. So 5 lichens belongs to same genus and different species. And among this Permetrema tinctorum abundant species, and isolated these 5 compounds from the species. And these 5 compounds we used as a standards. And this without doing an isolation, these 5 compounds were mapped their existence in the different the 4 species of the Permetrema. So for this, we developed we optimized a method of extraction and then LCMS conditions and mass conditions, chromatographic condition, mass conditions. With the optimized conditions, we developed a total ion chromatogram of 5 species, and then after analyzing the fragment ion peaks and molecular ion peaks, we have characterized the structures, almost 40 to 50 structures we have characterized from all 5 species of the Permetrema lichen. And then based on the fragmentation pathway, we also classified their category. These are dipeptide compounds. We also justified by proposing their fragmentation pathway. And similarly, dipeptidones also categorized another 10 compounds. And we also propose here the fragmentation pathway, these compounds, how these compounds are forming, by using these fragmentation ions and molecular ions. So these are the monophenolic compounds and paracoric type compounds. Finally, we concluded the study by distribution of these compounds, how these compounds are distributed among the 5 species, like whether these are specific to any specific species or they are distributed in the all lichens. So some compounds are specific in all species, but some compounds are the non-specific to on this particular lichen. So similarly, the same study we also did with Xylotum granatum and then Cypadaca bacifera. By using LCMS technique, we developed a total iron chromatogram and then identified the compounds by taking help of the fragmentations. And we also developed a extraction yield from the for the 4 compounds from the Coccolus hirstus, which is the one of the famous antiviral plant. And recently, Sunpharma is also working on the development of the anti-dengue formulation based on these 4 compounds. Here in our study, we have increased the yield of these 4 compounds, active responsible 4 compounds, by doing the different extraction techniques and developed the HPLC strategy and LCMA strategy. We also verified the marketed Ayurvedic formulations, whether these four compounds are existing in the formulations or not. So, and further, we also did the metabolite profiling by taking the help of mass fragmentation pathway and characterized those compounds with the help of the fragment ions and molecular ions. So, the quality control aspects we know pretty well. Today, the society is facing particularly the quality control aspects of the herbal drug. Here you can see this gentle lady hospitalized by taking the herbal supplement, which is known for the adulteration. So because of these adulterations and lack of quality control, herbal products are not becoming famous, and we need to do the standardization by checking the different aspects. And as we know, the compounds are specific to the region and genospecific and atmospheric specific and region specific. And we need to take the account of all these parameters while doing the standardization and doing the formulation. And also, majority of the plants are wild source, and the major compound, the therapeutic activity of the compound should be quantified from the formulation, like secondary metabolites like alkaloids and saponins and coumarins and flavonoids. And these are the different parameters which has to be looked while doing the standardization: physical parameter, chemical, biological, and botanical. So in the physical parameters, moisture value, ash value should be checked. And the chemical, the raw material should be free of the pesticidal residue and free of heavy metal residue. And in the biological, microbial contamination should not be there, and pharmacological evolution, toxical evolution should be there. And in the botanical, nowadays some of the companies are doing the replacing the original plant with the synonymous plants, where the original plant is scarcely available. So for this, we need to do the botanical, microscopic and macroscopic evolution of the plant to identify whether it is an original plant or synonymous plant. These are the analytical techniques well approved by the USFDA and USDA, HPLC, HPTLC, and UPLC, LCMS, and GCMS. And every technique has its own significance in the standardization of the herbal drugs. So we are also working in this area where this is one of the example where this Asuria lappa, one of the important medicinal plant, exists in the northern part of the country. And these 2 are the therapeutic markers, dihydrocortisolactone and cosnolide. So if you are making the formulation of the Asuria lappa, these 2 compounds must be present. Then only it is the genuine plant. So for that, we have procured the plant material, and these 2 compounds are quantified by developing the HPLC method. And further, we also screened some of the marketed products for the identification of these two existence of these 2 drugs. Fortunately, these 2 compounds are in the detectable limit in this formulation. Further, we also studied their degradation behavior and their stability studies, whether they are stable in the stressor environment or not. Fortunately, these 2 compounds are stable enough. So similarly, we also studied different Boswellia serrata formulations by identifying the 6 boswellic standards: keto boswellic acid, aceto-keto boswellic acid, different parts. These are the 6 marker compounds, therapeutic markers, well known from the different pharmacopoeia. and we have taken almost 30 to 40 products and then analyzed whether these 6 compounds are present or not. Only 3 to 4 companies are following the and having this at least 2 to 3 boswellic acid in their formulation. That is a pathetic situation we are facing, even though this boswellic saccharide is a very known for their arthritic pains used from the ancient times to till date, but the lack of quality control parameter is not getting famous. So I conclude here. I would like to thank my director, Dr. D.S. Reddy, and my guru, Dr. J.M. Rao, and other funding agencies for their generous funding to carry out my research. And not least, I am thankful to my students because of their hard work only. Just I presented their work in this forum. And I also thank the management of the Vishnu College of Pharmacy, Bhimavaram, particularly Dr. Nataraj Garu, for giving the opportunity to share our recent work. So these are the pioneered Indian scientists who dedicated their entire life in the cherishment of the natural product chemistry. Here we can see T.R. Sheshadri is one of the well-known natural product scientists, and T.R. Govindachari is a famous scientist who his entire life he worked on the isolation of and study of the neem plant. And Asima Chatterjee is one of the first women scientists who got the Bhatnagar Prize, and she worked on the indole heterocyclic compounds, heterocyclic nitrogen compounds. And Sukhdev recently passed away, and he is the father of glucurosterones. He completed 100 years of 100 years recently. I think another 6 months, recent 6 months back, he passed away. So, and again, I am happy to share that 2 of my products are in the market. And first 2 of my patents were commercialized to Sami Labs, Bangalore, and they made a product out of it, and Sabroxy tablets, which is known for its nootropic usage, and released in the USA for as a dietary supplement to combat the nootropics, neuropathy related, diabetic related neurological disorder. Similarly, we also worked on some NutraFoods, medicinal food plants, and we developed some 2 process technologies to combat the diabetes and anemia, and by using our traditional food plants. And in 2023, the product was launched by the Science and Technology Minister, Dr. Jitendra Singh, in ISCT under the name of Superfoods for You, as this technology was commercialized to Amaya Life Sciences, Hyderabad. And this product is now available in the Amazon, Flipkart. So these are the demonstrations where we demonstrated the product to the client, and these are the technology know-how while exchanging the technology. And this is the photograph taken while launching the product at ISCT. So just I would like to enlighten about the CSIR ISCT. CSIR ISCT is one of the premier lab in the field of chemical sciences under Council of Scientific and Industrial Research, which comes under Ministry of Science and Technology. We have 37 CSIR laboratories in the entire country, belongs to 5 disciplines: biological sciences, chemical sciences, engineering sciences, and information sciences, and physical sciences. In Hyderabad, we have 3 labs: NGRI, CCMB, and ISCT. ISCT comes under chemical sciences cluster, and NGRI physical sciences, and CCMB comes under biological sciences. So ISCT is one of the oldest lab, which completed 80 years of its service in the service of the nation and pharma for the development of pharma industries. And it is formed in the 1944 and established by the erstwhile Nizams under the name of CLSIR. And after independence, it was taken by the government of India and named as initially Regional Research Lab, Hyderabad. And in 1989, it was renamed as the Indian Institute of Chemical Technology based on the technology of different agrochemicals under the leadership of Dr. A.V. Ramarao. And in 2012, it was a CSIR was added since it comes under CSIR Council. ISCT completed almost 80 years of glorious years and contributed significantly in the building of the nation in the chemical and biological sciences. Almost our strength is our scientist strength, 180 scientists and 300 plus supporting staff. Almost 500 plus research students are there, and these are the list of patents every year we do. and we commercialize a lot of technologies to the different pharma industries. And these are the different divisions, starting from analytical and structural chemistry division, applied biology, chemical engineering, agrochemicals division, polymers and organic synthesis and process chemistry, natural products and medicinal chemistry, catalysis and fine chemicals, and energy and environmental engineering, oils and lipids. So our major strength is chemistry and chemical technology, and we also have the lipid science and technology, where we develop the the different process for original rich oils, rice rich oil, and we also commercialize the several technologies related to this. And we also have the agrochemicals and fluorochemicals laboratory, center of facility, and these are the different services we do, external services. NMRs, we have almost 8 NMR machines, and we also take some external samples for the analysis for doing the mass and SEM, TEM, and the other analytical service. And these are our recent contributions from IACT, Asia Technology, anaerobic gas reaction technology, from waste to wealth. We are generating the vegetable waste and food waste, from food waste to gas. We also install this bio plant in the Boyerpalli vegetable market, which is well appreciated by the Narendra Modi ji, our Prime Minister. And this is some of the membrane filtration water filters technology. We installed this water filters, water purifier plants in different fluoride affected areas, and also in Secunderabad railway station and Mumbai railway station, where water is produced from the atmospheric moisture, Megadut. So likewise, we also contributed in the COVID and by developing some of the technologies for favipiravir and remdesivir, and we also commercialize the technology of the one adjuvant and commercialize it to Bharat Biotech, which is part of Covaxin. So every Covaxin vaccine contains the ISCT molecule. So that is the recent contributions. So with this, I once again thank you for giving the opportunity, and I thank the audience for patiently listening. And I'm happy to answer any queries and any doubts. Thank you once again.
Thank you so much, sir. So, dear delegates, any questions are there? Can you please ask? Thank you. Dear delegates, any questions are there? Can you please ask?
Sir, about the herbal formulations,
Sir.
is it any possible techniques rather than the marker compounds? Is it possible to evaluate other components along with the marker components, sir?
Yeah, we can evaluate, sir. There are different markers called therapeutic marker, negative marker, and phytomarker. So therapeutic marker is the marker which, because of its presence only, that is responsible for the activity. And negative marker is the toxic compound.
Okay, sir.
And phytomarker is the compound which exists but with moderate activity or not active also. We can, but generally formulations are prepared by taking account of the therapeutic marker only.
What is the best method for, because markers generally, whereas synthetic compounds means we can get from the premier that qualities, but whereas like markers, where do we get this is?
Markers means I have some library of natural compounds, and
Yes.
we also supply to the different pharma, like natural product industries, like natural remedies, Himalaya, where they use only natural product based markers. And some there are some companies also which sell only pure natural products, compounds only for making formulations.
Thank you, sir. Thank you.
There are no questions, sir. Thank you, Suresh Babu, sir.
Thank you,
Thank you
sir.
so much. Hereby, I'm here for a vote of thanks. So my name is B. Brahmaya, working as assistant professor in the Department of Pharmaceutics. Good afternoon, everyone, and good afternoon, all delegates and principal sir and Suresh sir. It's great honor to propose vote of thanks on behalf of the Vishnu College of Pharmacy, Bhimavaram, on this 1 week online national FDP one challenges and innovative solutions in the drug discovery and development. I extend my heartfelt gratitude and thanks to our speaker, sir, Suresh sir, for enlightening our with so many overview of bioassays, guided approach, random approach, LCMS guided approach, and then all those reduction inhibition studies on 5 species. And then, so we have learned so many valuable insights with us, sir. Thank you so much, sir. And a thank to all the participants for all your active participation and active engagement. Thank you all, and thank you so much. So over to principal, sir.
Thank you, sir. Thank you. Thank you.
Thank you, sir. Thank you, sir. Thank you very much. Thank you, sir.
Hereby,
Right, sir.
thank you, sir. Hereby I'm ending the section, sir. Thank you. Thank you all. Thank you, Suresh, sir.